Caloric restriction, CR, the practice of reducing overall calorie intake without causing malnutrition, continues to stand out as one of the most robust and reproducible interventions known to influence lifespan and health span in a wide variety of organisms. From yeast and worms to rodents and primates, a consistent finding has emerged. When calories are restricted, aging slows, health improves, and in many cases, lifespan is extended. At the cellular level, one of the most compelling mechanisms thought to underlie these benefits is the preservation of telomere length, an essential marker of cellular youth and genomic stability. Research on CR has shown that it slows the rate of telomere shortening, a natural process that occurs as cells divide over time. Telomeres serve as protective buffers at the ends of chromosomes, preventing DNA damage and the loss of genetic information during replication. As they become critically short, cells enter senescence or undergo apoptosis, contributing to tissue dysfunction and age-related decline. By maintaining longer telomeres, caloric restriction appears to keep cells healthier for longer, delaying the onset of degenerative diseases and functional decline. The positive effects of CR on telomeres are thought to arise from its systemic impact on metabolic stress and inflammation, two of the most significant contributors to telomere attrition. CR reduces the production of reactive oxygen species, ROS, which are harmful byproducts of normal metabolism that can damage DNA, including the delicate structures at chromosome ends. In addition, it lowers systemic inflammation, as evidenced by reductions in pro-inflammatory cytokines and markers such as C-reactive protein, CRP. These dual effects of lowering oxidative stress and inflammation create an internal environment that is far more conducive to cellular preservation and genomic integrity. Furthermore, caloric restriction influences several key molecular pathways associated with longevity and cellular repair. It enhances the activity of sirtuins, a family of proteins that regulate metabolic efficiency, stress resistance, and DNA repair, all of which are crucial for maintaining telomere integrity. CR also activates AMP-activated protein kinase, AMPK, and downregulates the mechanistic target of rapamycin, mTOR, pathway, both of which play essential roles in cellular energy balance and the suppression of unnecessary cell growth. These molecular changes work together to slow the biological clock, reduce cellular wear and tear, and ultimately help preserve telomere length over time. Importantly, caloric restriction does not simply extend life by keeping organisms alive longer in a weakened or restricted state. It improves health quality across the lifespan. Animal studies repeatedly show that CR leads to lower incidence of age-related diseases, better immune function, improved insulin sensitivity, and enhanced cognitive performance. These effects suggest that CR extends the period of life spent in good health, what researchers call the health span, which may be even more meaningful than raw lifespan extension. Human studies, while more limited due to practical and ethical constraints, offer early indications that the benefits of CR may translate to our species as well. Participants in the calorie comprehensive assessment of long-term effects of reducing intake of energy study showed improvements in numerous biomarkers associated with aging and metabolic health, including reductions in inflammation, oxidative stress, and insulin resistance. While direct measurement of telomere length in CR participants is still an area of ongoing research, the reduction in factors that damage telomeres strongly suggests a protective effect. What's also encouraging is that CR doesn't necessarily have to be extreme to be effective. Even modest reductions in caloric intake, on the order of 10 to 20% below baseline needs, appear to confer measurable health benefits in both animals and humans. This makes CR more achievable and sustainable for individuals seeking to optimize their long-term health. Moreover, emerging strategies such as intermittent fasting or time-restricted eating may offer similar advantages by mimicking the beneficial stress responses triggered by continuous caloric restriction without requiring constant energy reduction. In the broader context of anti-aging and preventative health, caloric restriction is an elegant demonstration of how simple, non-pharmacological interventions can profoundly influence the biological aging process. It reminds us that the pace at which we age is not entirely fixed, but can be modulated through conscious lifestyle choices. By protecting telomeres, enhancing DNA repair mechanisms, and minimizing cellular stress, CR reinforces the idea that aging is not merely about the passage of time, but also about the state of our internal environment. As scientific understanding continues to evolve, caloric restriction remains a cornerstone in the field of longevity research, a natural, accessible, and highly promising approach to safeguarding the body's most fundamental systems. For individuals committed to maintaining vitality and resilience into older age, the evidence in favor of controlled caloric reduction is not only scientifically sound but profoundly hopeful. It represents a powerful affirmation that aging well is within reach, and that the most meaningful interventions may lie not in future technology, but in the choices we make today.